All right. Hi, everybody. Here is your long-awaited neuroanatomy video. Since, as you know, we didn't really have time to have everyone do this in person, I promised you long ago, beginning of this quarter, that I would give you some additional content by video. It's been a heck of a quarter. I don't know if you noticed, but because of that, it's taken me a while. But now, on this particular Friday night, I am going to record some content, and hopefully at least you will consider me a man of my word after this. Okay, so let's look at some different brains. I am here in my own lab, and I am showing you a collection of brains that I collected for you. As you may be able to tell, this is a human brain. So this one right here, the first thing we see is dura mater. Very thick, very durable. It's a tough mother, as they say. Here now we're looking at the exterior portion of the brain. Some things you can see is that there's another meninges, there's another membrane over these cortical gyri and sulci here. This is going to be your arachnoid membrane. You can tell that because there's some blood vessels that we can see here just underneath it. The arachnoid membrane is full of these blood vessels. Here's a portion of the temporal lobe that does not have any of that arachnoid membrane on it. It may or may not have the third meninge that you might be that is of interest, the pia mater. It is really kind of almost contiguous with the brain itself. You really can't see it except for under a microscope. So a couple other things we can see here on the exterior portion of the brain. This is a sulcus that is separating the temporal lobe from the rest of the brain, so from the frontal and the parietal and occipital lobes here, especially parietal and frontal. If we were able to, if the arachnoid membrane there wasn't there, I could pull this open and actually show you another piece of cortex which is just underneath here. You really have to kind of like cut it like this, something like that, in order to see it. But you would see another area of cortical folds that's deep inside of here. It's kind of smushed in there and then expands a little bit, about approximately to here. That's going to be your insular cortex. Okay, let's look at the bottom of the brain. A couple things we can see right away is here is your optic chiasm. These optic tracts would go up to the eyes. That's what they did before they were broken. You can also see a little hole here. That is going to be where the pituitary actually comes out. Let's see if you can, I don't know if I can focus on that real well or not. But the pituitary would have been kind of here. It was encased in bone, so when this brain was removed, it was destroyed. But that's what's going on there. You have a couple of big blood vessels here. Ask your nearest MD to tell you what those are. I don't know. Probably really very important though. A couple other things you can see: your brainstem. Here's your pons, which is the top of the brainstem. Midbrain is basically right underneath here. That's where your like kind of substantia nigra and whatnot will be. Let's see what else we can see. Your hypothalamus is going to be right underneath this area here. Your pituitary comes right out of the hypothalamus. That's where that is. Olfactory bulbs, which will come up again in a little bit. I actually don't know. I don't really see any olfactory bulbs on this particular brain. They must have gotten torn off at some time along the way. They would have been approximately right here. The reason I bring it up is there maybe like, let's say, they might be like something like right here is where they should approximately be. They're pretty small compared to the rest of the brain, but they do exist, which is in contrast to the next brain we'll look at. Other thing that's very prominent here is your little mini brain, your cerebellum. That's what we're looking at from the bottom. It's kind of like torn up a little bit, but that's what we got. Let's take it from the back. So this cut right here would have been from the autopsy when they actually removed this brain. They would have taken a big circular saw and cut the skull. A lot of times they aren't that careful. That's when you get this kind of damage. Let's looking at it here from the back. Let's pull the cerebellum down slightly. Got a lot of meninges in there, makes it kind of hard to see stuff, but you can see right here in your mid-sagittal sulcus. Not a lot to see in there really from the behind here with these meninges, with this big dura mater chunk here, but that's what we got. I was hoping we could see the colliculi. It doesn't look like they're really very visible here. They're probably under these meninges, so we'll look at that on the mid-sagittal cut brain. What else? So your hippocampus, if you're a hippocampophile, as a lot of people are here in this department, is going to be deep, so you can't see it. This is the exterior of the brain, of course, but if you went down about maybe a centimeter or so, it'd be approximately right here in your temporal lobe. Your amygdala is going to be right up at the front of that, right about here. Okay, so that's, I guess, the main stuff we can see on the exterior of the human uncut brain here. Let's take a look next at the mid-sagittally sliced human brain. So I guess we'll start on the exterior. We see a lot of the same stuff, these cuts from the autopsy. We see the temporal lobe, your frontal, parietal, occipital lobes. And as we look at then from the back, this is what I was, well, it's a little hard to see. I guess we can best see it from the mid-sagittal cut here. These little bumps right there, you guys all saw these, I think, in your sheep brain or your pig brain dissection. Those are your colliculi, so your inferior and superior colliculus have to do with sort of especially automatic auditory and visual responses. Other things that are really prominent here on the mid-sagittal cut are the corpus callosum. This is the biggest connection between the two brain hemispheres. There's also other ones. Here's one, the anterior commissure. We're going to see that in a moment in a chimp brain and in a cut brain. Other things, here's your optic chiasm. Your optic tracts would have come up here to the eyes. You can sort of see where the pituitary would have come off. You can't really see the hole here, but it would have come out right about here. Just above that is your hypothalamus. That's what we got right here in this area. Here's your thalamus, right above it, basically. And here's that pons that we saw from below. You got a lot of these cortical spinal fibers, a lot of also nuclei to do with respiration, all sorts of other reflexive, probably unconscious sorts of processes. You can see kind of this big white matter tract here. This is probably part of your cortical spinal tract. This will be coming from anywhere, like for example your motor cortex right around here. That's going to be sending down to the spinal cord and eventually to your muscles, anything about voluntary movement. So that big white matter tract is all those myelinated fibers from the cortex. Another thing you can kind of see is that if we look here kind of into the ventricle, so this would be a ventricular space here that would be filled with CSF. You can kind of see this little mass right here. This is going to be your basal ganglia, or your striatum, basically, caudate putamen. That's a little bit visible here from the midline view. If we followed this back down, this ventricle would kind of curve around and we would get back into the temporal lobe, as we saw from the bottom and the side of the brain. That's down to your hippocampus there. Here's your cerebellum, very obvious, very distinct structure. You get all these myelinated inputs. These are these big peduncles that are coming in from all kinds of places into the cerebellum. You have your more superficial nuclei of the areas of the cerebellum with your Purkinje cells and whatnot. You also have your deep cerebellar nuclei, which we'll be able to see on a coronal cut of the chimp brain in a moment, but they're way deep inside of there. They'd be like kind of right below here, I believe. And those are the big outputs back to the rest of the brain. What else can we see here that's of note? I think we got the main stuff from the mid-sagittal view. Oh, here's your cingulate cortex. You hear about this a lot, and especially in imaging studies, it seems to light up with everything. There's actually a funny poster that somebody did at Society for Neuroscience probably close to 10 years back now, saying basically the cingulate cortex does everything, and that was the SFN poster. It made a little bit of a scandal because it's kind of a joke, but you can look that up. I'm sure it's somewhere online. Visual cortex back here in occipital. You got your parietal and your frontal cortex from the midline here. Here's probably your ventral medial prefrontal cortex. Your orbital cortex is going to be down here, right up here in the front of the brain on the bottom, and your dorsolateral prefrontal cortex, which we talked a lot about in class, somewhere right around in this area. Okay, we've now done the human brain. Let's go to the next brain. So we got here a dolphin brain. You can see in comparison to the human brain, it's a little smaller. Dolphins are bigger than us, so already we've got some interesting information there about brain to body size. A couple things we can see about looking at the dolphin brain here from the top. Big old cerebellum. Maybe even bigger proportionately than you get in the human brain. Real big, a lot of cerebellar tissue. Another thing that really stands out is that compared to your human brain here, you've got these big chunky gyri, cortical folds. Here in the dolphin brain, much smaller. They're more tightly folded, basically. This is in some ways allowing them to have basically more surface area of cortex. If you were to take this whole thing and unfold it, you would, I'm not actually sure what the exact proportions are, but it's going to be kind of in the neighborhood of what the human brain has. It's more tightly folded in part This is partly a physical principle. So the dolphin brain has fewer layers than the primate brain, as like a human one here, for example. Since it's like it has fewer layers, it's almost like taking a piece of tissue paper. You can fold that, very sharply and get a nice clean fold. Whereas if you took a big old piece of cardboard, it's going to be a much more chunky fold. That's the same principle here. It's a thicker cortex. The cortical layers are actually thicker than in the dolphin. So you get these. You kind of you have to have these not quite so folded of a cortex here. Let's see. Let's turn the brain over here. One thing that doesn't quite stand out because our human brain didn't have olfactory bulbs either. Again, they would be approximately right in this neighborhood. Here in the dolphin brain, you actually don't have olfactory bulbs to speak of. They basically totally atrophied. If you think about what a dolphin is doing, is living in the water. There's not a lot to smell. They don't really have much of a sense of smell, if any at all. So those have atrophied. That's another little thing about function. One thing we can see, though, is an optic chiasm. Looks kind of similar to our human one in some ways. We get a nice desiccation here. The two eyes are coming in. Some of the information crosses, some of it doesn't. Same type of situation in a dolphin. Tells you a little bit of something probably about their world. They do use their vision quite a bit, just like humans and other primates. Here's your temporal lobes. Here's your frontal lobe. Where exactly the division between the frontal and parietal, for example, lobes are in a dolphin brain, I don't know. Presumably there's like a motor cortex and a sensory cortex, primary motor and sensory cortices. Here around somewhere in this neighborhood too. I don't know how to identify that in a dolphin brain, but I'm assuming that they work pretty similarly given that they're all mammalian brains.